Hello? Hello? Welcome to this webinar by the Massachusetts Consortium on Pathogen Readiness. I am Arlene Sharpe, Chair of Immunology at Harvard Medical School, co-director of the Evergrande Center for Immunologic Diseases at Harvard Medical School and Brigham and Women's Hospital, and co-leader of the MassCPR.
And I'm Bruce Walker, also co-leader of the MassCPR and the director of the Ragon Institute of MGH, MIT, and Harvard. And with Arlene, want to welcome you to this webinar on emerging innovations in COVID-19 therapeutics and clinical management. The MassCPR is barely 2 months old, but we're now up to about 500 members that cross the institutions in Massachusetts, including Harvard, MIT, BU, Tufts, UMass, the Brigham and Women's Hospital, Mass General Hospital, Boston Medical Center, Children's Hospital, Dana-Farber, Mass General, the Gates Medical Research Institute, and the Massachusetts Department of Health. We also collaborate with 2 institutions in China on this global pandemic. We have a number of working groups, and today the symposium is being sponsored by the Clinical Management Working Group and the Treatment Working Groups. And we have 7 different speakers representing different MassCPR institutions. And without much more ado, we'll get started with one final note. The work being presented today is mostly unpublished. It's not been peer-reviewed, and it contains preliminary data. And the meeting is being recorded and will be posted on our website for public viewing. So with that, we'll go ahead and introduce the first speaker, Hari Arthanari, who is at the Dana-Farber Cancer Institute and the Blavatnik Institute at Harvard Medical School. Hari, please go ahead.
Thanks, Bruce. The new coronavirus has an arsenal of proteins that help invade, infect, and attack our cells. Currently, there are no drugs that are specifically designed for SARS-CoV-2. Our mission here is to identify drug candidates in an expedited manner. The drug discovery has 2 wings. One is an experimental approach where a researcher uses the wet lab to find molecules that will kill the virus, similar to what Rob would be talking about. The slides are moving faster than I thought. Right? And the second is a computational wing where you can harness the power of computers to identify drug candidates. Here we can sift through about billions of molecules. This is also referred to as virtual screening or in silico screening, and this would be the prime focus of my talk. Can I have the next slide, please? In virtual screening, we pick a target site on the protein, take a candidate molecule, and place it in the active site of the target site, and computationally evaluate how best the molecule can fit in there. We try different poses of the molecule to determine what the best pose of the molecule in the pocket would be and determine a docking score. We repeat this for several drug candidates. And it's, you go back to the slide, please. We repeat this for several drug candidates to derive a list of high affinity binders, which will then be experimentally validated. This concept of virtual screening is not new. The next slide, please. Though, we should look at this with a new light today because of 2 seminal publications that appeared last year that showed that we can increase the potency of the drugs derived from virtual screening by dramatically increasing the number of molecules that we can screen. The slides are advising, yeah, which led to a News and Views article that says the bigger is better in virtual screening. And in order for us to do this for the COVID-19 protein, we need to screen through billions of molecules. But this provides a computational challenge. The next slide, please. On an average, it takes about 15 seconds to dock a molecule. And this would take about 475 years for us to dock 1 billion compounds on a computer. But for the COVID-19, we need to be able to do this in a matter of days. To overcome this challenge, the next slide, please, we designed Virtual Flow, an open source drug discovery platform that harnesses the power of computing clusters. Virtual Flow linearly scales with the computational resources that are available, allowing us to screen billions of molecules at an unprecedented scale. We decided to use virtual flow to take a multi-pronged approach to target proteins of SARS-CoV-2. Next slide, please. Here we target 16 proteins of SARS-CoV-2, and for some of these proteins, we target multiple functional sites, giving us ammunition to target the protein even when the virus mutates. Here I show you some examples of a few strategies that we use to target these proteins. The next slide, please. Here I show you the crystal structure of the main protease of the protein MPRO and an active site. Now we show a molecule that was identified by virtual flow that snugly fits. The slide is advancing faster than, yes, that snugly fit into the active site. And this molecule would have a binding affinity about low nanomolar. This is not the only molecule we have identified. From a screen of about 1 billion molecules, we have identified about 6,000 molecules that will potentially target this active site with a nanomolar affinity. Next slide, please. In a second strategy, now we show that we can target 2 instances of the same protein. Shown are the crystal structures of the same protein MPRO, the 2 independent crystal structures, and you can see the structure on the right has a deeper pocket that will be transiently available to engage ligands. This pocket is also called the cryptic sites, and using virtual flow, we have targeted both the pockets to identify ligands. The next slide, I'll show you that the cryptic site engages a ligand with a much higher affinity, and these could be much more efficacious ligand to target the main protease. And the next slide, please. Here I show you an alternate strategy of targeting the copier, which is the polymerase, the same protein that you'll see Bruce also talk about later. And here we are targeting this particular protein by targeting multiple functional sites of the protein. This particular copier, the polymerase, requires 2 helper proteins here shown in pink and blue. And what we're doing here is targeting both the active site as well as these additional sites that'll inactivate the protein. We go to the next slide. First, we show that how we target the active site. We can see a beautiful ligand that's bound to the active site. This is the same place the drug remdesivir binds. And our computation showed that our drug candidate is about 2 orders of magnitude tighter than remdesivir. In addition to the active site of the main engine of this protein, of the polymerase, the next slide, please, we also target the axillary sites, which is where the helper proteins bind. So any one of these 3 candidates should be able to inactivate the protein, and together all these 3 candidates should be able to more effectively inactivate the protein and be resistant to mutations. The next slide. In summary, we have targeted about 16 proteins with 33 target sites with a total of 35 billion docking instances using 75 million CPU hours at the Google Cloud in an unprecedented time of about 3 weeks. What this has led us is to have a set of highly potent hit drug candidates that can be now experimentally validated. The advantage of such a method is that we can get to this in a much faster scale, and targeting multiple parts of the machinery allows us to effectively combine these to provide cocktail options. And also some of these targets would be resistant to the mutations that the virus might do in the future.
Please.
The path ahead is we take the top hits, we account for dynamics on the receptor, and then we test these compounds experimentally in the platform that is set by the MassCPR. One is a platform that we will look for the ability of these drugs to kill the virus. And the second is a mechanistic aspect where we see if our drugs engage the target properly. With that, I'd like to acknowledge the team that is doing this work and the help from MassCPR. And I would like to introduce the next person, Davey, who's actually going to test these molecules in a cellular context.
Thank you, Hari. So the big question is, how do we test all these therapies that Hari and the speaker after me, Joe, and other scientists are wanting to know if they stop the virus infecting cells, right? So screening molecules is hard to do at best of times. It's made even harder when we're dealing with a dangerous pathogen like SARS-CoV-2. So my lab has been working for testing molecules against Ebola virus, which is obviously a dangerous virus. And we developed techniques to try and be quicker, cheaper, and better at identifying treatments for dangerous virus infections. Next slide, please. So I work at the Needle, which is a lab in Boston that has BSL-4, which is the highest level of containment facility. It has the lab shown in the middle there, has 12 specially designed airlocked laboratories where we can do drug testing and trying to understand how the virus functions. And on March the 19th, on the right, you see myself and a postdoc, Hiroyuki Mori, going into the lab with our first sample of SARS-CoV-2. That was March the 19th. Since then, we've developed a high throughput screening method to rapidly evaluate small molecules, antibodies, peptides, and other treatments to try and stop infection. So, you know, the objective for us was to test thousands of such treatments very quickly, and then try to get them as rapidly as possible to efficacy testing in preclinical models. And I do some of that, and I'm also helped enormously by Anthony Griffiths, who has got a great preclinical team. Next slide, please. So here is the process in a nutshell. So on the top left there, you see we're using plates that have 384 little wells inside them, right? So one plate, we can test 384 different types of drug. And this is a hurdle when you're working at high containment, how to deal with that sort of scale. And then we can take many of these plates every week, hundreds if we need to, and then put in them, you see up the top, you put cells, they start out being blue. You add the drug, you add the virus, and then wait a while, a day, and then detect infection of the virus using an antibody which binds to the virus proteins. This is a very similar antibody which we'd find in patients after they've recovered from virus. Anyway, the antibody binds to the virus and tells you how efficiently those cells are infected. So the plot in the middle shows you a number of little dots, right? So each dot is a different drug test that we've done. And on this particular plot, we have 2,000 of them. And if you look on the y-axis, you'll see we're looking at how many cells were infected by using that antibody detection system. And on the x-axis, we're looking at cell number. Now, it's important to do both those things because you might stop infection, but unfortunately, your therapy might be detrimental to the cell. And so when a therapy is detrimental to the cell health, the number of cells drop. So things on the lower left corner there, that little red dot that I put over the blue dot shows you a lack of cells. There's not many blue spots in that image from our microscope. If we go up to the top right there, you'll see there are many little blue dots, which each blue dot corresponds to a cell. And there are many green dots around those blue dots. And each one of those is an infected cell that's expressing the virus protein, right? And so in that case, that particular treatment did nothing to stop the infection. But on the lower right, which is the most exciting part, of course, is you'll see that there are many blue dots, which means the cells are healthy. And you'll see there are very few green dots, which means there is very little virus infection. So, and this is as I said, exciting because this is a candidate for a potential therapy treatment. Next slide, please. So what are we doing? All right, so one of the largest screens we've done so far is working with Mark Namchuk, Flo Wagner, and myself. It's a collaboration between Harvard, Broad Institute, and Boston University. And we've taken 6,800 drugs from the Drug Repurposing Hub from Broad Institute, and we've evaluated them for blocking infection of virus in that system I showed you in the previous slide. We're also evaluating some unique drug libraries. One is from John Porco at Boston University, which are large type molecules that block virus proteins interacting with host proteins, which is a good candidate for therapy testing. We're taking candidates from computer-based screening methods. Joe Loscalzo will talk to you about that after me. We're taking candidates from Hari Arthanari's 10 billion compound computer screen, which he showed you before. We're looking at peptides, which can interfere with entry of viruses into cells. And we're also testing thousands of other small molecules that have been donated from industry, academia, and just the average day person on the street. And the unique thing about what MassCPR is providing is we're providing a conduit for people to submit their potential candidates and have them tested in our system quickly, so we can rapidly evaluate what's going to work and what's not going to work. Next slide, please. So a big problem with, why is it taken relatively short time, but actually, people want to have an answer quickly, sooner, for testing things. So how can we be quicker, and how can we find drugs for new viruses we know nothing about? This technique here shows you that we can detect the virus genes themselves instead of the proteins. So the green protein I showed you before with an antibody, now we can get away from relying on antibodies, and we can detect the virus genome itself, which is the pink stuff. And this is quick and efficient, and we can get from saying the sequence of the virus to doing a screen in 2 weeks. Next slide, please. So in summary, we can test thousands of therapies a week. We are working with many people, Boston, Massachusetts, and nationwide. MassCPR has been very generous in supporting this effort and is helping us to do this efficiently. Candidates that you might want to have tested, please look at the MassCPR website, and also Dr. Mark Namchuk is organizing that testing. And of course, thanks to the people in my lab who have made this possible, and our collaborators on the right across the city and the state. And that's all I have. And so the next speaker is Dr. Joe Loscalzo, who will be talking to you about using artificial intelligence to try to identify new candidates for treatment.
Thank you, Rob.
The Harvard.
Well, for the past 15 years, our group has been working to develop the new field of network medicine, which combines network science and systems biology in order to understand the complexities inherent in human biology and disease. Today, I will share with you our network medicine-based approach to drug repurposing for COVID-19 treatment. Next slide, please. This slide illustrates our principal tool for analyzing biological complexity. The fuzzy background in this slide on the left represents the protein-protein interaction network. Remember that proteins are macromolecules that are very sticky. They associate with each other in highly specific ways. On average, each protein in a cell associates with 10 other binding partners. This interaction map, or interactome, comprised of nodes or proteins and edges or specific binding interactions, illustrates how extraordinarily complex the landscape of protein interactions is in a cell. We have a wide range of quantitative approaches that allow us to analyze and exploit this complexity. And among the results of these analyses to date is included the key observation that the proteins underpinning each specific disease are highly localized within this interactome, comprising disease modules or subnetworks, examples of which are given in color in this slide. These can be used to explore specific disease mechanisms and identify specific drug targets. Next slide, please. Here I illustrate two network-based approaches to identifying drug targets. The first, on the left, is the conventional strategy. By standard reductionist experimentation, one identifies a target in vitro, then scans the library of drug compounds to which an individual or a company have access for those with the proper binding properties that modulate the target's function in the right way. And then it's moved to candidacy development. On the right is a different strategy that we developed specifically for repurposing approved drugs. We first identify the interactome-based disease module of interest, shown in pale blue or gray, and then identify targets protein targets for approved drugs for any other disease that may be localized either within the disease module of interest or near it, that is within a couple of binding interactions, and elevate its candidacy. Next slide, please. Turning to SARS-CoV-2, the situation becomes more complex as illustrated here. We now have three related interaction networks to consider. The viral proteome on the left, the drug disease or drug target module on the right in blue, and between them, the host, human target proteins that interact with the viral proteins and are near, by, or equivalent to the drug targets. We denote this central module, the Covidome. The next slide illustrates the real Covidome derived from our use of Gordon's identification of 332 human protein binding partners for the 26 SARS-CoV-2 proteins that he expressed as bait proteins in his system. We filtered this list of proteins to include only those that were expressed in the lung. That turned out to be about 215 or 65% of the 332. It's important to point out that the benefit of mapping these expressed host proteins to the interactome to create this real-world Covidome on the left is that the interactome provides the missing protein links that consolidate and define the associated disease module and identify pathways in their near entirety that may regulate pathogenicity and may be good targets for drug intervention. Next slide, please. We next analyze the Covidome for potential drug targets within it or in its proximity within the global protein interaction network. Without going into mechanistic detail, we developed and applied three analytical strategies. The network proximity strategy shown on the top, which I reviewed 3 slides ago, a network diffusion strategy shown in the middle, and an AI-based neural network, graph convolutional network strategy shown at the bottom. The pipeline numbers refer to different quantitative variations on each strategy that we utilize for developmental purposes. As a result of this analysis, next slide please, we developed a combined or aggregated ranking algorithm for 7,000 FDA or EMA-approved drugs and examined the top 10% in detail, curating them to yield the 81 candidate drugs shown here for further study. This table includes some drugs already in clinical trials, highlighted in green with a deeper color indicating a greater number of trials, as well as other drugs that have recently been shown by conventional experimentation to have antiviral activity in vitro, such as praziquantel and nitric oxide donors like nitroprusside. These associations lend one level of validation to the analysis. More important, however, is the range of drugs not previously known or hypothesized to be useful in this disease, which warrant further analysis. Importantly, we cannot tell from this analysis, one, whether a candidate will suppress or promote infection, which is where Rob's assay becomes so critical, or two, whether a candidate will have other side effects exerted via another target in a different part of the global interaction network. For these reasons, all candidates must be evaluated first in vitro and only thereafter in patients. Next slide. So next steps include performing the in vitro analysis with these in silico candidates with Rob Davey and updating the analysis regularly as new data become available. Please note the link to the archived manuscript for those of you who wish to learn more about the methodology and read more about the curation approach. In the last slide, I wish to acknowledge my colleagues who developed the methods and performed the analyses shown here. My thanks to them for their creative work performed rapidly in the heat of battle, and to you for your attention. So our next speaker is Richard Kaufman from Brigham and Women's Hospital, who will discuss plasma therapy.
And I just might interject that for those people participating in this call, please use the Q&A function for questions that you may have, which we will come back to at the end of the talks. Richard.
So thanks very much, Bruce and Joe. So I'll be talking about convalescent plasma as a treatment for COVID-19. May I have the next slide, please? So convalescent plasma, or plasma that's obtained from people who've been infected with, in this case, SARS-CoV-2 and then recovered, is a form of passive antibody therapy. And the idea is really simple. So essentially, you take antibodies that have been formed against a specific pathogen, and you infuse them into someone that's infected. And here, I'm showing some antibodies that are binding to and attacking SARS-CoV-2. And some antibodies can not only bind to a virus, but they can also block infection of the virus. And these are called neutralizing antibodies. And it's thought that antibodies that have this neutralizing function may be the ones that are of greatest benefit to patients. So the approach of using convalescent plasma has been done for many, many years. And I would say that the data are limited and conflicting. But convalescent plasma has been used in recent epidemics like SARS-1 and MERS and Ebola, and it's being tried now for COVID-19. May I have the next slide, please? So I'm going to talk about two uncontrolled case series from China. And these are really the only data that we have so far that's been published on the safety and efficacy of convalescent plasma for COVID-19. So in a series that was published recently by Shen and colleagues in JAMA, 5 patients with severe COVID-19, all of whom were on mechanical ventilation for acute respiratory distress syndrome, were all treated with 400 mLs of convalescent plasma. And here are some of the data. So day 0, indicated by the red arrows, is when convalescent plasma was infused into these patients. And the first thing that you can see in the plot on the left is that the levels of neutralizing antibody went up in each of the patients by day 1. So that's what you would expect if, in fact, you're infusing neutralizing antibodies in the convalescent plasma. So then the question is, what happens to the patients? And so in the plot in the middle, you can see that oxygenation improved in the blood of all 5 patients by day 12. And in the plot on the right, and again, each of the colored lines represents a different patient. After getting convalescent plasma, 4 of the 5 patients defervesced, their fevers went away within 3 days of receiving convalescent plasma. And so there did seem to be some efficacy in this series, potentially. And I can tell you that 3 of the 5 patients were able to get off of mechanical ventilation and were discharged home. At the time this paper was published, the other 2 patients were still in the hospital in stable condition. But the main limitation of this sort of data is that there were absolutely no controls. So we don't have any patients who were similar who did not receive convalescent plasma. And we actually don't know what would have happened to these patients had they not received it. May I have the next slide, please? So this is the second published paper. Again, this is an uncontrolled case series from China. In this case, there were 10 individuals, all hospitalized with COVID-19 and ARDS. 3 of the 10 patients were on mechanical ventilation, and all of them were given a single dose of convalescent plasma. In the column that says neutralizing antibody titer baseline, what that is showing is the amount of neutralizing antibody that was available in the patient's blood before being transfused with any convalescent plasma. And you can see in the column next to that in blue, the numbers that are bolded indicate that half of the individuals showed an increase in their neutralizing antibody titer. The next 2 columns are looking at RNA that was in the sera of these patients. 9 of the 10 had positive detectable SARS-2 RNA. And then after receiving convalescent plasma, all became negative. All of these patients were said to have improvements in things like cough and shortness of breath within 1 to 3 days of receiving the convalescent plasma. But here again, the problem is that there were no controls. May I have the next slide, please? So there are some risks of giving convalescent plasma. So one is lung reactions. And this is true not only of convalescent plasma, but really of any plasma transfusion that may be given to a patient. One reaction is called TACO, transfusion-associated circulatory overload. And this is basically giving too much fluid to a patient. Often this would be someone with a bad heart or bad kidneys, and some of the fluid can back up into their lungs and reduce their ability to oxygenate. And a second important lung reaction is called TRALI, or transfusion-related acute lung injury. This is an inflammatory reaction that can happen that affects the lungs, and again, can cause difficulty in breathing in patients that have this. There are often allergic reactions that happen after patients receive plasma. Many times, what happens is just the patients will get hives and itchiness, but sometimes patients can have anaphylaxis, and this can also be life-threatening. There's a low but not zero risk of transmitting an infection like HIV, hepatitis B, and hepatitis C, and so on. These are really, really low risks, but not zero. And then finally, there's an idea that, and some data to support this, that if you were to passively transfuse just the right amount of antibody, it might actually cause a worsening of a viral infection. By coating a virus with specific antibody, the virus may be more actively gobbled up by cells, and this may actually worsen an infection. And so all of these have to be considered as potential downsides of using convalescent plasma. And so we think at this point, there's what we would call equipoise. That is, there's uncertainty about the risks and benefits of using convalescent plasma to treat COVID-19. May I have the next slide, please? And so our group—
Please wrap up.
I'm sorry?
Go ahead.
Our group, as well as many others around the country and the world, are beginning to study convalescent plasma in trials. This is a study called the ESCAPE study, which stands for Evaluation of SARS-CoV-2 Antibody-Containing Plasma Therapy. We plan to enroll 220 hospitalized COVID-19 patients at the Brigham and Women's Hospital and several other hospitals in Boston, including Mass General. And these patients will be randomly assigned to receive either 2 doses of high titer COVID-19 convalescent plasma or regular plasma. This study is being done in a blinded way, so neither the patient nor the caretakers will know which group the patient is in. And an important feature of this trial is that, in contrast to many other studies that are being done of convalescent plasma, we're only collecting convalescent plasma from individuals who've been proven to have high titers of neutralizing antibodies using a high throughput assay being done by Mike Siemens' lab. He's also a part of MassCPR. May I have the next slide, please? The primary endpoint of the ESCAPE study is something called a WHO modified ordinal scale. This has been used for other studies in this area, basically of antivirals. Individuals at baseline will be scored according to what's shown here. So a score of 0 means you have no clinical evidence of infection. A score of 9 is death. And the scores in between represent different levels of illness. When we enroll patients in the ESCAPE study, they'll be in the categories that are shown by the red box. So 3, 4, 5, or 6. So the most well ones will be hospitalized, but with no oxygen requirement. We will also take people in level 6, intubated, but with what we would call a mild acute respiratory distress syndrome, and patients in between those. And essentially, at day 14, we're going to measure in the 2 groups whether people get better or worse according to this scale. And we will also use as endpoints things like 28-day survival, viral clearance, and some other key endpoints. And so let me just go to the next slide. And so I just want to acknowledge many people that have helped me with this study, in particular, Lindsey Baden and Cliff Moe and Mike Seaman and Stephen Piantadosi. And at this point, I'd like to say thank you and move on to our next speaker, who is Wayne Marasco, who'll be talking about next generation human antibody discovery, insights from SARS, MERS, and SARS-2. Thank you.
Thank you, Rick. Next slide, please. So, developing antibodies during a viral outbreak has some major limitations to it. It really involved the limitations in obtaining raw material to be able to do this kind of work. In particular, obtaining blood samples from infected patients as a source of B cells, which are antibody-producing cells, really is problematic because of a number of reasons. There are government restrictions. This was clearly the case in the SARS outbreak in China in 2002, 2003. as well as the MERS outbreak that occurred in the Arabian Peninsula in 2011 and 12 that was centered really in Saudi Arabia and Qatar. Very difficult to get samples out at that time. There are also public health issues that are both local, regional, and global. There are a number of regulatory issues, and really there are national concern issues. So is there another way? Next slide, please. So we really developed the idea of using large libraries from healthy individuals where we would isolate their B cells, randomly recombine the heavy chains and light chains of their antibodies, and display them on the surface of a bacteriophage, which is a virus that infects bacteria, that allows us to link the surface antibody to the gene that it carries, and therefore becomes a microbial B cell. And using this technique, you can do a lot of rapid discovery. Next slide, please. So this is an example of what has happened over the field of developing this technology. When you start with libraries that are small in the million range, you get many antibodies to different proteins on the surface, but they tend to be low affinity in the 10 to the minus 6 to 7th range. As the libraries increase in size, the 10 to the 8th, the libraries become more efficient, your affinities increase. By the time you get into the billions and tens of billions of members, you now have antibody libraries that are quite robust. Their affinities are down in the single picomolar, single nanomolar and picomolar range, and you can use these as a rich source of developing antibodies. I built a library in 1996 with 57 healthy volunteers. I took 20 mLs of blood from each of these individuals, and from that we built a 27 billion member antibody to phage library. Next slide. And basically in the SARS outbreak, we really developed, showed that you could do this quite efficiently. The left is a crystal structure of one of the antibodies that we pulled out, and the spike protein is on the bottom, the antibody is on the top. And you can see compared to the right side, which is ACE2, the receptor, and the spike protein on the bottom, that that antibody on the left overlaps precisely the same region that the spike binds to ACE2, and thus this antibody was able to potently block virus infection, validating the fact that out of these libraries you could pull out antibodies against infectious agents for which the members had not been exposed. Next slide, please. So we did this again in the MERS outbreak, and this time we were more sophisticated in what we were able to do because we were able to get a lot more antibodies out, and we used these actually to map the interaction surface between the spike protein on the right side on the bottom and its receptor for MERS, which was human DPP4 on the right. And we were able to use a series of neutralization escape mutants that we developed on the left to be able to find which antibody bound to which region of the spike, and if the virus tried to escape, we would find out exactly how it would do that. And by using a combination of antibodies then in a cocktail, we were able to block virus escape pathways, and thus the whole idea of using antibody cocktails came into being. Next slide. Next slide, please. So we've used this technique now to in the SARS-2 outbreak, and I just wanted to briefly show you how this is done on the slide on the left. So you put your antigen, your spike protein in the tube, you coat the tube, you wash the tube, you pour in your library of phage, you let all the unbound phage wash away, then you remove the antibody bound to the tube, you infect bacteria, you plate the bacteria, and this cycle goes on and on. And every time you do, you get about 1,000-fold enrichment of antibodies to your target in the original library. And if you do it 2 or 3 times, you can get 1 million or 10 million-fold enrichment of those rare binders that bound to your target in the billions of members that were there. Next slide. And this is what we obtained from these efforts. So for those that are interested in the scientific details, we used a competition assay shown on the top. If it was non-competing antibodies, they're on the top sensorgram or competing on the lower one. The middle is just a checkerboard. If one competes antibody against itself, you knock out binding. And this allowed us to map the regions that the antibodies were bound to. And the bottom shows the result of all that mapping studies. we essentially found 3 groups of antibodies that are grossly put into bin 1, bin 2, or bin 3. And bin 1 is directed to the S1 protein, bin 2 is directed to the spike, and to a region outside of the receptor binding to the interface of ACE2, and bin 3 are ones that directly block by blocking the ACE2 binding site. Next, please. And with this, we were able to pull out a large number of high affinity, broadly neutralizing antibodies shown here. This is our bioassay for it. It's a neutralizing assay. As the antibodies decrease in concentration, the antibodies start to lose inhibition, but you can see in this there are several at the very lowest concentration that give 100% inhibition of binding. Next slide. So in summary, the next generation antibody discovery can be used to isolate therapeutic antibodies to emerging viral pathogens. It takes less time. The antibodies are high affinity. They're of high specificity. It allows us to take the findings from convalescent plasma that Rick Kaufman talked about and have confidence that next generation antibodies can be developed into therapeutic agents. And there's really a tremendous amount of knowledge to be gained by comparing the antibodies that come out of next-gen antibody discoveries with those that occur naturally, particularly for vaccine designs. And with that, I'd like to go to the last slide and thank the members of my lab that were involved in this work, as well as the support by MassCPR that has made all this possible. Next slide, please. And I'd like to now introduce Boris Juelg, who will be talking about his work on viral polymerase.
Thank you very much. I want to show you now a couple of slides on potential targets, drug targets, and in particular medications that are available or have been available already that can be reused now for testing against SARS-CoV-2. And I'm going to walk you through an image, and you've seen that before. That's the viral life cycle, and Harry had showed a similar image before. And what you can see here is pretty much the life cycle of SARS-CoV-2 from the infection part on the top, the fusion where it enters the cell through the ACE2 receptor, then the translation part, proteolysis. But then an important step for the viral replication is step 4, as you can see here, translation and RNA replication. And there are several enzymes that are involved in that, and these enzymes have been target for drug development in other viruses, but also obviously for SARS-CoV-2. Virus then goes into packaging and then is getting released from the cell. And all these different steps pretty much are potential drug targets. And a couple of ideas or drug ideas are labeled here. And we just heard about the monoclonal antibodies, neutralizing antibodies, which potentially would block the virus from entering the cell. But other agents like lopinavir, ritonavir here in the middle of the slide, which is an HIV drug, which can prevent the proteolysis step. So there's different ideas out there. I'm going to target now specifically the viral replication step. Next slide, please. And I'm going to talk about the viral polymerase. And so the viral polymerase is a very important enzyme which is responsible for copying the genetic material of viruses, therefore essential in the life cycle of viruses. And there are different agents which have been developed actually to inhibit this polymerase, in particular nucleoside analogs. And next slide, please. So these nucleoside analogs target the, as I said, RNA-dependent RNA polymerase. And here you see the SARS-CoV-2 genes that make proteins which are relevant for the viral life cycle. In particular, there are several non-structural proteins and enzymes which are proteases, but then the polymerase and viral helicases, for example. And there are other structures and proteins like the spike protein, what we just heard, which is the target for antibodies. Next slide, please. So this is also an image which you've seen before from Hari, which is the crystal structure of the RNA-dependent RNA polymerase. And there's this drug binding pocket, which is highlighted here in the pink purplish color. And in that pocket, actually different agents, different nucleoside analogs can tightly bind. And by doing that, they pretty much inhibit the polymerase and therefore inhibit the virus to replicate. Next slide, please. And what we see here now is some data for one of those nucleoside analogs. It's called remdesivir. It's a drug from a company called Gilead, and you might have seen that in the media already because it got a lot of attention. So this was an agent which was out for several years already, had been developed initially and tested for Ebola, and was now redeployed actually to be tested for SARS-CoV-2 because it had quite strong potency in vitro in cell culture assays against the virus. And so the study that was published was done in China, was recently published in The Lancet, which compared remdesivir versus control. As you can see here, the red and blue lines are pretty in parallel. And overall, they had to stop the study early because they didn't have enough patients anymore after the Wuhan pandemic pretty much subsided. But they could not see a statistically significant difference in terms of improvement rates between those two arms. As I said, the study was ended early and they didn't fully enroll, so their power to really detect differences went down. So therefore the result is not completely clear. However, the NIH had a randomized controlled, placebo-controlled blinded study, the ACT study. And although there's no data published yet, there was a press release on April 29th, and Tony Fauci from the NIH actually reported some positive results showing that there was a faster time to clinical recovery, 11 days versus 15 days in the remdesivir treated individuals. And so that was significant. However, they did not see a real difference in the mortality, although there was a trend maybe, 11% mortality in the remdesivir treated individuals and 11% in the control individuals. So the jury is out there. We don't have seen the full data, and that was only a prelim analysis. But so Gilead is doing a similar large study as well, testing remdesivir. And so there's a lot of data still coming and a lot of analysis have been done on remdesivir. Next slide, please. Favipiravir is another agent, also a nucleoside analog. It's approved in Japan. It's an anti-influenza medication, actually. And there have been 2 trials where there's data available, both done in China. One study on the left, you can see they compared favipiravir against another anti-flu medication, Abidolol. And they did not see a difference here in time to recovery, clinical recovery, again, comparing those 2 antiviral agents. So there was no placebo control involved here. However, on the right side was a second study where they compared a group of individuals treated with favipiravir versus a group of individuals treated with the HIV drug lopinavir/ritonavir. I mentioned that before. And there they saw a statistically significant difference in time to viral clearance, 4 days for favipiravir and 11 days for the HIV medication lopinavir/ritonavir. And they also saw that on the chest CT findings, there was an overall better improvement for the favipiravir treated individuals with 91% versus the HIV medication treated individuals with 62%, and that was significant. So this agent is being tested in several studies across the world, including in Boston. Next slide, please.
Please wrap up.
Several other polymerase inhibitors are out there. I don't want to mention them all. Overall, what those graphs show you is that the potency of some of them, HSV medication, pencyclovir, ribavirin, require much more drug actually to inhibit virus, at least in a cell culture assay. And so the jury is out if these should be tested, and some of those studies and trials are open. Next slide. The studies currently, favipiravir is enrolling at UMass, Brigham, and MGH. This is the first US-based study actually testing that agent. It's a small study with a target of 50 people, so we hope to enroll that fast. Remdesivir, as I said, NIH study had the first results completed at MGH. However, Gilead has several studies still on with BIDMC and Brigham and other sites across the United States. Next slide. So at the end, I want to acknowledge Robert Finberg, Nicholas Issa, Lindsey Baden, helping with the favipiravir study and all the clinical trial teams and the co-I's and funding. And this was made possible by the MassCPR. Next slide. So next, I want to introduce John Stone, who's going to talk about blocking cytokine storm in COVID-19 pneumonia. And John is an expert in IL-6 antibody treatment. So John.
Thank you very much, Boris. So I'm going to talk about whether we can take an existing medication, a monoclonal antibody, and prevent the cytokine storm, which is talked about so much now in the scientific literature and in the media. Next slide, please. So this is a conceptually bold approach because it proposes to use an immunosuppressive medication, an IL-6 receptor blocker, to prevent the cytokine storm. We hypothesize that this approach may be successful in this moderate, intermediate stage of the disease, in which the disease begins to affect the lungs. The viremic stage, in theory, is beginning to wane, and at this point in the intermediate stage, the host, the patient's inflammatory response takes over, causing enormous and potentially fatal tissue injury. Next slide, please. So on the left is a chest X-ray of a healthy individual. On the right is a patient with severe COVID pneumonia. At this point, the moderate stage has progressed to severe. Both of the lungs have very diffuse inflammatory infiltrates, pneumonia, and the patient is intubated on a ventilator. This is the stage that we want to present by interrupting the disease earlier. Next slide, please. So cytokine storm is not a new concept. It was not invented when it came to COVID-19. It's actually been discussed for many years now in the context of other diseases, adult onset Still's disease, macrophage activation syndrome, and others. All of these highly inflammatory conditions are linked, in theory, by interleukin-6, which is a key inflammatory cytokine. And we hypothesize that this is a major driver of cytokine storm, not only in these other diseases, but in COVID-19. Next slide, please. So COVID-19 has been, IL-6, serum concentrations of IL-6 have been shown to predict respiratory failure in COVID-19. This is a study from Germany, released only several weeks ago, and patients' baseline admission IL-6 concentration predicted those who would progress to requiring mechanical ventilation. Next slide, please. COVID-19 is a highly hyperinflammatory state, and we must acknowledge that there is more than just IL-6 elevation going on. IL-6 elevation is shown in the graph on the upper right. The red curve shows patients who died of their infection, and the survivors' serum IL-6 concentration much lower are shown in blue. But IL-6 does not necessarily tell the entire story, and other inflammatory mediators are shown on other graphs here. D-dimer, serum ferritin concentration, and LDH are also elevated strikingly in these patients and also correlate with survival as opposed to death. Next slide, please. So tocilizumab is a monoclonal antibody which binds to the IL-6 receptor, and it outcompetes this inflammatory cytokine for its receptor. IL-6 concentration levels in the blood soar after patients have been administered tocilizumab, but the cytokine is rendered physiologically inert following the administration of tocilizumab. Tocilizumab has been available for about 10 years and is highly effective in rheumatoid arthritis and giant cell arteritis and other conditions in which IL-6 is an important mediator. Next slide, please. So we have developed a trial known as the Back Bay trial. BAC stands for Boston Area COVID Consortium. The trial is comprised of 7 centers in the Boston area, and most of these centers are also represented in the MassPCR. These are the 7 centers here, and if Mr. Ragon is in attendance of the symposium today, he'll be pleased to note that all of these hospitals, except one, use Epic for their electronic medical record, and that indeed has been instrumental in conducting this trial rigorously, with the ability of coordinators to remote into patients' medical records through Epic, even if they're at a different hospital, and assess for adverse events. Next slide, please. So the Back Bay trial is a randomized double-blind placebo-controlled trial. Patients with moderate COVID pneumonia at risk of progression to the severe stage are randomized in a 2-to-1 ratio to intravenous tocilizumab or placebo. Patients have their investigational medication administered within 2 hours of signing the consent form. The major inclusion criteria, 2 out of these 3 are required. A patient must have a pneumonia, COVID pneumonia, must require supplemental oxygen, or have had a fever within the preceding 72 hours. And they also must have evidence of a highly inflammatory state in their blood. Next slide, please. The goal of enrollment is 278 patients. The primary outcome is a very hard outcome. It is the prevention of mechanical ventilation, the prevention of progression to respiratory failure. There are a number of other important secondary outcomes that have been discussed by previous speakers and are very similar. Next slide, please. And here is our status at the moment. This slide is a little bit out of date. We've actually enrolled at least 117 patients as of this morning. We are moving very, very quickly. The goal is to fully enroll the trial by the end of May. And if I could have my next slide, please. I would just like to thank all of the Back Bay trial investigators at these 7 Boston area hospitals. Thank you very much.
So thank you, John. My name is Raj Gandhi. I'm one of the co-leads of the clinical management working group, along with Lindsey Baden. And it's a real pleasure to have organized, along with the Therapeutics working group, this exciting symposium today. I think you've seen we've gone from computational biology to clinical trials. Over the next 30 or so minutes, we'll be taking questions both from panelists as well as from attendees. And so if you do have a question, please use the question and answer function in the portal to ask your question, and we'll try to get to as many questions as we can. I'm going to kick things off with a question for our last 2 speakers, for Dr. Boris Juelg and John Stone. Boris, you showed us that we were successful with HIV and hepatitis B and hepatitis C, in large part because of the advent of combination therapies. In those cases, many times targeting the replication machinery or the copying machinery of the cells. I'm curious what you think about the prospects for combination therapy for COVID-19. Do we need to target several parts of the virus, or does this virus mutate less than other viruses, and will that not be necessary? And then I'd like you and John to have a bit of a back and forth as to whether we need both immune blockade for the cytokine storm and antiviral therapy. So go ahead, Boris, and then I'd like you to weigh in, John.
Well, it might be a little bit too early to say yet, given that we're so early in the readout of the initial clinical trials, and to see actually which of those antivirals have good clinical efficacy. And I showed those graphs. You can see some in vitro activity against viruses in a cell culture, but it doesn't always translate right away in a better outcome in a patient. So I think we really have to wait for the data from those initial antiviral drug trials to get a better idea if that's sufficient or if it's insufficient. And if you think about influenza, you think about the Tamiflu drug, which is very commonly used. The effect there is also not 100% switching off the virus completely, but really reducing the virus amount and improving the time to clinical improvement by just a day, and still we use it and it's clinically beneficial. So the question is, where do we want to get? Do we want to have a full switch off of the virus? Then most likely one of those drugs alone will not be sufficient, and we need maybe combinations. Or we wait for some of all these new agents that are getting currently discovered, as we've seen by Hari and others. There's a lot of different prospects, and they might be much more potent actually in doing that.
Maybe John, can you comment on, you alluded to at the beginning, the approach of turning off this overexuberant immune system. How is that going to interplay with a need for viral control, and how do you view these modalities in terms of how we're going to get to where we need to be with COVID-19?
So Raj, I think still early in this recent epidemic, we're still learning how to do trials and how to study this disease. I do indeed think that combination therapy of antiviral agents plus anti-cytokine agents may be the best approach going forward. I think these approaches would be tolerated well. I note that in one of the early remdesivir trials, mortality was reduced in the antiviral approach, but I think it was by 3.6%. So there's certainly room for improvement there potentially, and an anti-cytokine therapy, particularly if started earlier before patients are intubated, may really help move the ball down the field there.
Okay.
If I may just follow up on that, Raj, this is Lindsey to John and Boris, and Raj, you as well. Does the timing matter? If someone is mildly symptomatic at home, is that different than if, is the inflammation an intrinsic early parameter, or might it be a later parameter that has a pathogenesis, and it depends where in illness the individual is, where we deploy treatment? What are your thoughts?
I think it absolutely does, Lindsey. There's so many factors, comorbidities and timing of illness that determine whether a patient progresses to respiratory failure or not. I really do feel that intervening earlier is likely to be more successful. So early in the moderate stage may be even more successful.
One comment I'll make in that regard is that remdesivir, which we've heard a lot about, is now being thought of in a second trial to be combined with an immunomodulatory drug. So that approach of combining different arms of targeting the virus and preventing overexuberant immune response is going to be explored. And I'm sure there'll be exciting data coming out of that. I'm going to ask Mark and Jonathan if they can comment and lead the discussion around some of the other aspects of today's program. Mark and Jonathan.
Yeah, I can chime in. I mean, I had a question for Hari about, I think one of our goals ultimately would be to have drugs that don't only work against SARS-CoV-2, but perhaps have activity against SARS-1, against MERS, and other emerging coronaviruses perhaps that we haven't met yet. So my question, Hari, is how are you thinking about sequence space around those proteins and those enzymes? And are you incorporating in your pipeline computationally, perhaps looking for broadly active agents?
Great question, Jonathan. And as you know, there's fair amount of conservation between the different coronavirus families, and the target sites that we are targeting are fairly conserved around SARS and MERS, with the exception of probably the spike protein and its interaction with the ACE, which is mutated. So by this, and if we combine this with the notion that the virus has a proofreading ability and it doesn't mutate as rapidly as other viruses, I think there's a fair amount of conservation on that drugs would be pan-active against other species of coronavirus.
Thank you, Hari. And I had a follow-up related question for Wayne Marasco. Question would be that, there are bioarchive now preprints showing perhaps that the virus, as it's spreading across the globe, is acquiring mutation in the spike protein. And so I was wondering if you could comment perhaps on how you could incorporate that in your antibody design, again, with the idea of having broadly active agents. You mentioned the MERS example, which was great, but are you thinking about those mutations as well for SARS-CoV-2?
Oh, we're absolutely thinking about them. I mean, this is part of the discovery process. When you try to target the pathogen itself, you always have to take into consideration that there's ongoing virus evolution. So some of those, the mutations that have been reported concurrently, several of them are in the receptor binding domain, but outside of the ACE2 binding site. One of them is in the N-terminal domain. You've got to really choose your epitopes wisely so that neutralization is a range of activities. You've got to make sure that you're targeting a region for which the virus cannot undergo neutralization escape, or if it does, it's at a significant cost to viral fitness. So part of the discovery effort in doing these kind of studies is really ranking the neutralization activity, not only on its potency, but its ability to prevent neutralization escape and drive viruses, if they try to escape, down a dead-end evolutionary pathway. So it's very much part of the discovery process.
Okay, great. I'll pass it on to Mark now to see if he has any questions.
Yeah, I had one follow-up question for Rob Davey, actually. There's a lot of published literature out that's starting to emerge around different methodologies for doing in vitro and repurposing work, but inconsistencies are starting to emerge between those data sets. So where would you guide in terms of trying to set up the most robust way to do initial discovery, and what would you want to add as the platform gets going to continue to improve the validity of what can be done in vitro to predict potential winners?
Yeah, I think the biggest issue right now is the virus is actually tricky in terms of what cells it will infect robustly, and therefore you can screen efficiently. And so at the moment, many people are screening things in Vero cells, which are a monkey cell line. Of course, we're similar to monkeys, but we're not the same. So, you know, it's very important to do follow-up studies in human cell types. And we've got some of these human cell types to work fairly well. One is a liver type of cell line. Another one is actually lung epithelial cells, which of course that's what we care most about, but they're difficult to work with. And finally, we're also trying to do follow-up assays in primary lung systems. These have been used in industry as well as academia, and they're sort of coming to the fore now. So I think they're the things that are going to show the most promise in validating these primary assays that everybody is using.
And then maybe one other question for a couple of the panelists, both our clinicians and our discovery scientists. We do make a lot of comparisons to influenza, and one of the things that is sort of an open question is this whole point about time of intervention in acute viral infections. Tamiflu works very late in the viral replicative cycle and also has a relatively narrow window of intervention where it has clinical utility. Maybe starting off with Boris or John, and then Rob, if you wouldn't mind jumping in. Is there any guidance you could give from a clinical or biological perspective of what's most likely to be the best way to intervene in this sort of an acute infection, but has a little bit longer course? Mechanisms we could look for, things that we can learn from, say, influenza that we should avoid.
Any thoughts, Boris?
Yeah, sure. I mean, if we can find something which blocks the virus from infecting anything in general, then that would be maybe the best step. You know, I mean, that would be that we don't have the virus infect any cell, and we don't have to then intervene later, kind of, would be ideal. Otherwise, you know, as we learned from influenza as well, the question is how much viral replication inhibition do we really need? You know, do we have to completely knock it out, or is it enough if we take it down a log of copy numbers, kind of, and maybe that's sufficient then for the body to take control? And as with influenza, the early phase of the infection is where those drugs do the most and are actually indicated first 48 hours in influenza, for example, in symptoms, for example. And it might be the same actually now for SARS-CoV-2, you know, something which you can do early to just take down the virus a little bit.
So Mark, in our trial, we're going to be measuring serial viral titers to see what happens to patients after IL-6 receptor blockade. In theory, anyway, it could prolong viremia and maybe lead to a worse outcome. If that's the case, then Certainly, I think we'd all feel more comfortable if we had an effective antiviral agent on board first or simultaneously. But we'll need to synthesize the data from all of the trials that are ongoing right now to figure out the next best approach.
At one point before our discovery scientists opine on this important matter, Boris, you mentioned early treatment. Obviously, in some other viral diseases, we have antivirals that work as prophylaxis or preventative therapies. So that would be of interest, of course, with SARS-CoV-2. Is there a way to prevent disease if you interfere early enough? One of the questions also related to mutational rate. Maybe you can just comment on what your perspective is on this virus's ability to mutate. This has been a point of discussion recently about, is this virus changing and mutating? How does it compare to influenza? How does it compare to HIV in terms of its ability to mutate? And then I'm going to let Mark's.
I mean, I'm not a virologist, so maybe there's others who can answer that better. But if you look at Betty Korber's recent data that's online, that's available, I think that's not peer-reviewed and published yet. But it seems like that there is some mutation activity going on. It's definitely not. It seems not as fast and as an influenza or definitely not HIV. So, gives us a little bit more time maybe to act there. But it's not completely silent either that virus doesn't mutate at all. So there are newer agents also in the nucleoside analog spectrum from the Barrett group, for example, that was published in Science Translational Medicine recently, which seem to actually be active even in some of the mutated forms of SARS-CoV-2. That's all in vitro and animal data so far, so that has not been tested in a clinical setting. So maybe people should think about that. Also, obviously, we would like to have antivirals which not only work for SARS-CoV-2, but maybe like 3 to 10 and MERS and all the others as well. So we'll see.
I mean, if I can add to that, from the virology standpoint, so with influenza virus, the RNA polymerase is thought to be more sloppy from the standpoint that it has a smaller genome size, and from the standpoint of SARS-CoV-2 with a genome size of about 32 to 35 kb, sloppiness would come too much at a fitness cost. So the virus actually encodes its own exonuclease that helps edit out mistakes so that genome replication is a more faithful process. And so presumably there should be less mutations, but it's not a DNA viruses, which those are the viruses that have the lowest rates of mutation. So I would say probably less likely than flu, but still possible, particularly given the size of the pandemic.
And flu has its multi-segmented genome, which makes it much more adept at generating new types of virus. And HIV can do recombination between its, RNAs, duplicated genome. So, but this virus is not in that category. So I think it's less likely to have such a mutation rate.
I think that was all the questions that I had. I don't know if Bruce, you were going to be able to go through some of the questions that were coming in from the audience, or did you want
to?
us
I think we can all do that. Here's one from Jeremy Luban, who's also one of the members of the MassCPR. If one had a promising anti-SARS-CoV-2 drug ready for clinical testing, what is the current capacity of MassCPR for accommodating new trials, recruitment of patients, et cetera? I guess that would be for Lindsey or Raj, maybe as heads of the clinical management working group.
Lindsey, you want to take that one?
I mean, I can say that there are an enormous number of clinical trials that are now ongoing as a lead-in to Lindsey's answer. Lindsey?
Sorry.
Lindsey?
Oh, yeah, I'm sorry. Can you hear me? So
yeah, now we can.
Yeah. So the question is therapeutic strategy.
Right, the question is about the number of, if somebody had, if somebody had a drug, a cytokine inhibitor, antibody, whatever, what's the current capacity of MassCPR to accommodate new trials?
Well, I mean, I think that what's been remarkable over the last month and a half is the number of, unfortunately, the number of patients who have been ill and the number of trials that have been launched. And so the capacity has been quite substantial. And across our institutions, there's tremendous enthusiasm and integration of effort, as evidenced by the different talks from Boris and Rick and John about studies going on across our institutions. The challenge, so in that sense, we have capacity and an incredibly integrated effort, will be the number of cases. The hope is through control measures, we won't have any more cases. So that will diminish the ability to do studies. But as long as there are cases going on in Massachusetts, we are well positioned to study them all across our institutions, including in Worcester, which unfortunately right now is really struggling.
I would just add that I think reaching out to us as the purpose of MassCPR is to foster these type of collaborations. And so I think Lindsey and I and others are more than delighted to talk about ideas that people have. I think that's why so many of these trials got launched so quickly, is that kind of collaborative spirit.
So I have a question for Richard about the antibody-dependent enhancement, which has been raised as a concern. I don't think that there are data that are definitive out there by any means, but it's a concern for vaccine development. Do you envision that the studies with adoptive therapy with convalescent plasma will help to answer that question? You're on mute.
Well, I hope so.
And what's your impression of that? Do you think ADE is a real concern?
I think it's a legitimate theoretical concern. I'm not aware of any specific data for SARS-2, but for example, there have been in dengue some signals of ADE being a real thing within human studies, and within some preclinical models in animals as well, using different viruses. It does appear to be an actual effect. And so I have no idea if this is going to be important or not for something like convalescent plasma. I think that though that we are going to have to look for it. And additionally, we expect that other passive antibody therapies will become available as early as this summer, things like hyperimmune globulin. So for convalescent plasma, we're just talking about literally getting 1 or 2 units of plasma from a donor and using a bag of that as the treatment. It's possible to pull together hundreds or thousands of plasma donations and make a much higher level of antibody. So it'll have to be looked at. And then of course there are recombinant antibody therapies that will also become available later this year. And I think it's theoretical, but we have to look.
And sort of at the opposite end of where you are with taking antibodies that somebody's immune system has produced, what Wayne has been doing is creating entirely new antibodies from people who've never seen the virus. The question to you, Wayne, is do you have potential candidates to move forward therapeutically, and what do you envision as the timeline for that? Do you think this is, the question is, how soon are we going to have something that really works? And can you also envision helping to prove that eventually a vaccine may be possible by showing that highly targeted antibody can actually impact the infection?
Yeah, Bruce. So the antibodies are really, they're in the early stage of development. Some of them are now in the hands of Anthony Griffiths down at Needle, who are doing neutralization assays, and we're arranging for animal studies to be done now. If everything holds up, at least the first tier of eligibility studies, which would be neutralization and animal studies, will be completed. And I think it probably is going to take a cocktail. I mean, given the fact that there are mutations that are out there, I would really want to go into the clinic with probably a cocktail of 2. But to answer your question, how quickly it can be developed, it really depends on whether the FDA would give emergency use authorization, because we know from DARPA projects that I've been involved in that you can produce kilogram quantities of antibodies in about 3 months. But if we have to make a packaging cell line like normally is done for conventional monoclonals, that's 9 to 12, 14 months. So it really depends. I mean, we could have them ready for the fall if there's a need. Otherwise, they're like vaccines. They're probably going to be ready in the spring of next year. But to the point of your second question about vaccine development, I do think they're going to be very important in guiding the way to vaccines. It also addresses the question that you asked, that Rick was responding to about antibody-dependent enhancement. So I think there's a few different questions. I know we're going to work on FC engineering with Galette. And, you know, there are certain things you want to protect against that potentially happening. So the short answer is it can be done by the fall if there's laxation in the regulatory approval process. Otherwise, they wouldn't be ready until the spring of next year.
Mm-hmm.
Thank you. I have a few questions. There's for John. The first is related to IL-6. Are there any data suggesting that lower expression of IL-6 receptor or IL-6 across patients is associated with decreased risk of severe disease?
No, there are no data to my knowledge. There is considerable speculation, though, about maybe more IL-6 receptors in patients with COVID, pneumonia with severe COVID, and about even higher circulating IL-6 concentrations than seen in other diseases in which tocilizumab is an effective therapy. So it speaks to one of the uncertainties about studying this disease now. What's the right dose? We are using the highest dose of tocilizumab used in other drugs, in other diseases. We hope that that is sufficient. We think that it probably is, but we don't know for sure.
Thank you. And in terms of cytokine storm, are there other cytokine clinical trials that are ongoing or being developed in addition to IL-6 related ones?
Certainly, as I pointed out, there are some other cytokines that are elevated in this condition. We see, for example, IL-1 may very well be a driver here. Some patients have extremely high ferritin concentrations. And in other diseases marked by a very high ferritin concentration, IL-1 receptor blockade is an effective therapy. The question is, will IL-6 receptor blockade be sufficient for that? It is in some other diseases. Whether it is would be as effective or more effective than IL-6 receptor blockade than IL-1 receptor blockade in this disease, we don't know. It's certainly another, IL-1 is certainly another potential target, as is GM-CSF, theoretically another target. There is a hyperinflammatory state in these patients. It is truly impressive. So one could postulate these and other things that might be worth testing as well.
And Arlene, there are at least a half dozen other pathways being targeted in clinical trials currently, from corticosteroids to more targeted pathways, as John has discussed.
Thank you.
There's another question from my former mentor, Marty Hirsch, or one of my co-mentors, talking about the great progress in HIV and HCV with combination antivirals. Are you studying any of your promising compounds in combination? It is not too early as we compile patient data to gather these in vitro data using typical studies of synergy from the earlier days with HIV and HCV. And I certainly remember the exciting days in Marty's lab when the discovery was made that combination therapy actually was incredibly potent against HIV, and that was in vitro. Who wants to take that?
Can I give a shot with the in vitro side? Combination therapy, of course, is the holy grail if you can get synergy. Additive effects are still good. So we're going to probably take probably the top 10 or so treatments that we get out of this and do them combinatorially in vitro and see what happens. Of course, you know, you can amplify side effects by having 2 drugs in the mixture instead of 1, and we're very aware of that. But we're going to give it a shot.
Maybe Bruce, if I could add one thing, because I know Joe had to jump off of the call. That's one of the big advantages, these sort of networked approaches too, is you could rationally think of unrelated targets that you would try to combine in vitro as well. So I think as more of these computational approaches mature and get access to larger data sets, I think that'll be really good fodder to go back to the idea of being able to look at combinations of, say, a host target with a direct antiviral where there's some synergistic response based on either the protein map or some other guiding principle.
And Bruce, I think along those lines, which Boris alluded to, is in the in vivo model, when we treat patients, what are the markers of success? And if it's going to be the WHO 7-point scale, which is incredibly crude, or do we develop biomarkers like an HIV viral load that allows us to see in vivo activity? And I think that's another area that is under active investigation that hopefully will help speed things up once we can figure out which biomarkers matter.
And I think your colleague, John Lee, actually has a promising looking assay, and as these get more sensitive, detection in blood is certainly I think a reality.
Can I just say something about the biomarkers in general? I mean, it's definitely super relevant, obviously, to measure our clinical outcome with our interventions. But I think one critical piece is as well, if we could determine who's at risk for more severe disease, and not just based on the risk factors of age and, you know, comorbidities and all that we kind of know now from epi data, but is there something we can, you know, measure at baseline that gives us an idea who's going to have a worse disease than others? Because then we could, you know, as we said, intervene much earlier, maybe with some of the anti-inflammatory agents already, to really prevent that, and wouldn't have to give it to everyone. So could really have a personalized approach with these different therapies. And I think that's hopefully the research will show now, given that a lot of those patients getting sampled for specimens that, you know, this immunology, virology, and so on is going to look at that and correlate that to clinical outcomes. And hopefully is going to come up with some signatures that help us determine and predict what the disease outcome will be in different people. And then we can act on that specifically with our interventions.
I mean, is IL-6 one of the things that is potentially a marker, John?
I think it certainly is, but it exists in a sea of other things that might be markers. And I think the predictors, we shouldn't ignore the very basic epidemiology of this. I'm reminded of the early days of HIV, when the epidemiology, who's at risk for the disease, were so important and so fascinating. And here we have obesity and hypertension and diabetes, and I mean, I've made the observation that there's no such thing as a skinny COVID patient. Obesity is a big issue here. These patients have BMIs of 38, and that's got to be figured into any sort of risk profile, I think, along with the things that we can measure in the blood.
And do you have any sense as to why obesity would impact this?
I do not, but it's a question worth giving a lot of thought to.
There's a lot of inflammation in obese patients, and so that'll be interesting to understand and follow up on as well, to understand what the elements are there. Well, I think our time has unfortunately come to an end, but on behalf of MassCPR, I would like to thank Jonathan, Mark, Lindsey, and Raj for organizing this timely symposium, and all of our speakers for their wonderful presentations. In this symposium, we have gone from molecules to clinical trials. We've heard about innovative strategies for identifying small molecules and repurposing drugs to treat COVID-19. Hari discussed his incredibly rapid computational screens to identify small molecules. Joe Loscalzo shared his innovative strategy to identify using network theory and AI to create the Covidome and then identify drug targets for repurposing. and these candidate molecules and drugs are being evaluated for their ability to inhibit SARS-CoV-2 through screens in collaboration with Rob Davey, who is rapidly testing an impressive array of molecules. We also heard about harnessing the power of the immune system to treat COVID-19. Richard Kaufman is partnering with Lindsey Baden and Michael Seaman to soon start a COVID-19 convalescent plasma clinical trial, as well as work from Wayne Marasco, who's now developing next generation antibody therapy and has potent candidates for therapeutic antibodies. John Stone discussed therapy for cytokine storm and a clinical trial of anti-IL-6 that will be conducted in 6 weeks. Truly unprecedented. Boris Juelg discussed clinical trials for drugs targeting the viral RNA-dependent RNA polymerase that are rapidly enrolling. This rapid progress is truly inspiring. And I would like to highlight that a number of these partnerships were created at the inaugural MassCPR meeting on March 2nd and are already bearing fruit. This illustrates the value of collaboration and how collective effort can catalyze innovation and progress. Finally, I want to thank our Dean, George Daly, for his support, enthusiasm, and inspiration, and our incredible leadership team, Willie Lynch, David Golan, David Huang, Lauren Hecht, Andrea Chen, and others for the tremendous work that enable these symposia. And finally, thank all of you on Zoom for participating.